right, good afternoon, ladies and gentlemen. We're going to talk photosynthesis today. This is part one. This, again, just like cell respiration was kind of a long set of notes, this is a long set of notes. So we'll do part one today. Hopefully you've watched the cell respiration ones. All right, so what we have been focusing on up to this point is how living systems, process energy. We talked about how the energy has been stored in glucose molecules, and we can gradually pull the energy off of that to make our ATP during cell respiration. How does the sun's energy get stored in the glucose molecule in the first place? And that's what we're going to look at today, photosynthesis. So this should look a little bit familiar. This slide is very similar to the slide that was on the cell respiration slide. In that slide, we were talking about, we showed like a gorilla or a chimpanzee, I can't remember, was eating some food. So how do we make that food, or how are plants and organisms that have chloroplasts making that food? So this is the same diagram we had in cell respiration, and we're just reminding you of the reciprocal nature about how chloroplasts are making, as a waste product actually, but it's a very useful waste product, they're making oxygen, and then purposefully, that's a funny way of saying that also, making organic molecules like glucose, and we're going to follow glucose specifically. But then those molecules are brought into the mitochondria, where we can make ATP, which is our short-term energy, and use that to flex our muscles or to cause our hearts to beat. And then it produces, as waste products, a little bit of water and carbon dioxide, which amazingly are used directly by the chloroplast in the reciprocal relationship, in the reciprocal process. So that's a super cool coincidence is the wrong word, but super cool the way they work together. So it says there's a reciprocal relationship between chemoheterotrophic nutrition and photoautotrophic nutrition. A couple things to keep in mind that can throw students off. Chemoheterotrophic is referring to eating. Photoautotrophic is referring to making food within the body. And plants do that's a funny way of saying that. I was going to say plants do both. You and I only do chemoheterotrophic nutrition. Like, that's the only way. We cannot store our own food or make our own food within our bodies. What I'm trying to say is we only have mitochondria. Don't forget that plants have both chloroplasts and mitochondria. So they're doing both of those processes. All right, moving on. And then the inputs of one are the outputs for the other, which, there we go, explains this curious fact that these two reactions are the exact opposite of one another. Please take time to look at these reactions. See how there's glucose here, C6H12O6, and oxygen. That's the products of the reaction of photosynthesis. And notice how those are the not products, but reactants. There we go. Brain was on a little delay there. Those are the reactants in cell respiration. All right, moving on. So plants are what we often think of as the things that photosynthesize. They are not the only photoautotrophs, but everything has to have a chloroplast in it, or has to be like a chloroplast in order for that to happen. So purple sulfur bacteria, cyanobacteria, unicellular algae, they have chloroplasts within them. And then interestingly, it says seaweed. That is in fact kelp. And just a little FYI, kelp is not a plant. It's an underwater organism that's in the protist kingdom. So it's with single-celled organisms that live in water. But it also has chloroplasts that can photosynthesize. All right, so in plants, photosynthesis happens in the leaves, which are organs specialized for this process. I'm going to show you my hand here. Pretend that my hand, there we go, pretend that my hand is a leaf, and then I'm going to turn the leaf this way. Imagine if we cut through my hand this direction and you were looking right at the cross section of my hand. Can you imagine that the bones and the blood vessels of my hand would look like circles? Well, that's what they've done in this diagram here, and I want to make sure you're really clear on that. I'm going to make this, I wonder if I can make it bigger. Oh, I can. There we go. So here's a vein within that leaf. So if you cut the leaf this direction, the veins would look circular. If they had bones, they don't. It would look circular also. Okay, so now that you understand what you're looking at, the top layer here, those are called epidermal cells. So that's like the top layer of my skin. Inside are the mesophyll cells. There's other cells, but we're just going to go with mesophyll for right now. And they contain the chloroplasts. And then there's veins that can carry water and sugars. And then on the bottom of the leaf is also epidermal cells. Notice they don't have any chloroplasts on the top or on the bottom, it's on the inside. But here, very importantly, notice that's where the stomata are located. You guys did a question in the last unit that had some stomata on it on one of your AP exams. So stomata are the holes, the pores on the bottom of the leaf that open and close to allow carbon dioxide in and oxygen out. The problem with stomata, the issue why they open and close, is that sometimes water escapes out of them during very hot or dry circumstances, and so they close to limit how much water gets out. Well, that's a problem because during photosynthesis, we need to be bringing carbon dioxide in and we're making oxygen. And so it screws everything up if those stomata are closed. So the plant is constantly adjusting based on how hot it is and how dry it is and how much water is inside the plant. If enough water leaves the plant through the stomata, it desiccates, which basically means it dries up and dies. So that's why it's got to moderate that. The stomata also, interestingly, are located on the bottom side of the leaf because that's more shaded. So the sun is beating down on the top of the leaf, and the bottom of the leaf, you can imagine, is shaded more. So that's the cooler side of the leaf. So less water is going to evaporate out of the cooler side of the leaf. So that's why the stomata are located on the bottom. All right, and then let's also take a look at the chloroplasts themselves. So you know that the chloroplasts are green. The reason that they're green is that they include a pigment molecule called chlorophyll, and we're going to talk about that in some detail right now. But let's take a look here at a close, can we pinch it close? There we go. So let's look at this chloroplast. First of all, remember that chloroplasts were believed to have been bacteria. They have a double membrane. And then, so here's the inner and the outer and the inner membranes. And then they have these green things on the inside. So any one of these discs is called a thylakoid. And then they have stacks of these discs. So imagine like stacks of coins. Those stacks are called grana. And the other, granum is singular, grana is plural. The other word that I want to make sure you know is the word stroma. Stroma is the liquid inside of the chloroplast. So it's kind of pointing to nothingness right there, and that's the stroma. All right, moving on. In chemistry, we've talked a little bit about light, but maybe not quite enough. So let's go into a little bit more explanation about light and how light works. Remember that light is a form of electromagnetic radiation. It's produced by the movement of electrons between orbitals. So if you're like, what's that again, KJ? Remember those box charts that we had when we had 1s2, 2s2, 2p6, 3s2? Do you remember all those? Those are the orbitals. And when an atom absorbs energy, say a photon, it excites its electrons. It can excite an electron from a lower energy level or a lower energy orbital to a higher one. And then when it returns back to its natural or neutral or not excited state, it releases a photon of energy. And we might be able to see that photon if the wavelength is within the wavelengths of visible light. We might not see that photon if the wavelength is much greater or much less than what our eyeballs are able to detect. Visible light is just one tiny slice of the larger electromagnetic spectrum. All right, so now chlorophyll is a pigment. What is a pigment? It is any molecule that interacts with light energy to produce a color. We're going to learn more about this as we go, so just bear with me. Chlorophyll comes in two main varieties, chlorophyll A and chlorophyll B. And I can't remember, nope, it doesn't close up. So let's close up on this image here really quickly. So this is chlorophyll A and chlorophyll B. They're very, very similar. The only difference being right here in chlorophyll A and B. You don't need to know that at all. But I just wanted to point out that each chlorophyll molecule has a magnesium atom, which is a little unusual, has a magnesium atom right in the middle. And that's going to be the source of our electrons. It's going to be ejecting electrons. And that's why the chlorophyll molecule ends up being really important. So we'll come back to that. While chlorophyll is the main photosynthetic pigment, and that's why plants look green usually, it is not the only pigment found in chloroplasts. There are other pigments that are called accessory pigments. We'll mention a couple of them here, the carotenoids and the xanthophylls, but there's even others. They absorb different parts of the electromagnetic spectrum. So we're basically, the leaf is trying to, the chloroplast is trying to absorb as many possible wavelengths of light as possible. Chlorophyll absorbs really well in the red and the blue range, but doesn't do very well in the green range. So there's others that are there to try to help absorb as much of that sunlight as possible. All right, so why are plants green? This is a little bit counterintuitive. Again, chlorophyll, which is the primary pigment in chloroplasts, absorbs light very, very well in the red range and in the blue range. But it doesn't absorb light well in the green wavelengths. So instead, it reflects green light back to your eyeballs. So it's a weird thing, but it's absorbing other colors and reflecting green. So a lot of students assume that it absorbs green light, and it's exactly the opposite of that. And we're gonna show you some cool experiments with that. So it says sunlight contains almost all wavelengths of visible light. Chloroplasts do not absorb all wavelengths of light equally. When plants are exposed to light, chloroplasts preferentially absorb light in the blue and red parts of the spectrum. Chlorophylls have an absorption spectrum. We're going to look at that in just a second. So that's what part of the spectrum they can absorb that is highest in blue and red, but terrible in green. The accessory pigments expand the useful range of light, called the action spectrum, which we're going to see in just a moment. But green light's still the least useful, and that's why plants look green, is because that light's being reflected back to our eyes and not being absorbed by the plants. So now, this would be super easy for students to gloss over and be like, oh, blah, 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 whatever, I don't need to pay attention to this. And I would actually argue that this is the kind of thing that AP is going to write a test question about, and we definitely don't want to scoop over it, or scoop. That's not a word. We definitely don't want to glide right past it. So let's talk about how do we detect what the absorption spectrum of a given pigment is. So look at these experiments, and then we'll talk about the Theodore Engelmann experiment, which I think is super cool. So first of all, on the technique, we're looking on the left side of the diagram right now. Imagine white light. Now, it could be just a light bulb giving off white light, or it could be the sun giving off white light. And we're going to run it through a prism, which is going to spread the wavelengths out into their different into the rainbow, into the form of a rainbow. I'm not going to go into the details there. And then notice the black thing. That is a filter, and that filter is only allowing light through that we want to allow through. And so right now the slit is set up in the first one. The slit is set up to allow green light to pass through, but it's blocking all the other colors of the rainbow. So green light is passing through, and then we're transferring it through a chlorophyll solution. So the solution that has this chlorophyll pigment within it. On the other side of the test tube, we have a device that is capable of detecting light and converting the light into an electrical impulse. Then when that's called a photoelectric tube. The galvanometer is capable of registering that electric charge and showing us how strong the electric charge is. So if more photons are being detected by the tube, the galvanometer will record a high level. And if not very many photons are being detected by the photoelectric tube, then it's going to show low. So when we have that filter set so that only green light is passing through, notice that green light not only passes through the filter, but it passes all the way through the chlorophyll solution. It's not being absorbed by the chlorophyll solution. It's being detected by the photoelectric tube, and we see the galvanometer recording a high level. Now we change in the second image, we change where that slit is, and this time we only allow blue light through. Blue light is one of the light colors that is good for the chlorophylls, so it absorbs. So when we go to pass it through the test tube that's full of chlorophyll, it absorbs that blue light. Very little light passes through to the photoelectric tube, and it's registered very low on the galvanometer. All right, so now let's take a look at how that is actually registered so that we have a graph of that. So on the vertical axis, we see it says absorption of light by chloroplast chloroplast pigments. So the higher it is, the more it absorbs. The lower it is, the less it absorbs. So let's just look at chlorophyll B, for example. So with chlorophyll B, I'm going to run my cursor on it. Chlorophyll B's peak, where it absorbs the best, is somewhere between 400 and 500. So we'll say 460 nanometers. Now, if you're not familiar with the prefix nano, it means 10 to the ninth. So 10 to the ninth meters is a nanometer. So very, 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 very wee. So that's what it absorbs best. Now, as we continue to follow chlorophyll B along, we see that it suddenly plummets when we get into the green area of the spectrum. That means it's not absorbing much of that light. And then when we go back into the redder area, we get a second peak, not as high as the first peak, but it does absorb some of the reddish orange light. And you can see that with the other ones as well. Okay, so that's the absorption spectra. We say it's plural because there's several different elements, different molecules that we're looking at there. Now, if we combine all of the different accessory pigments together, and we look at how well does a leaf or a chloroplast absorb, then that's called the action spectrum. And so because of the accessory pigments all combined, we have a nice peak in the purple and blues, but we still don't do very well in the greens. And that's why your eye sees it as green, because all that light is not getting absorbed. It's being reflected back to your eye. And then again, it absorbs well going down into the red area of the electromagnetic spectrum of the visible light that we can see. Now, the last part I think is the cool part and definitely is an AP type of question. I'm going to read the paragraph that's at the bottom and then help you understand it. The unequal utility, oh my gosh, KJ, of different wavelengths of light was first noticed by Theodor Engelmann. So what he's saying is that not all wavelengths of light are equally as good. He's the first dude to notice that. So he observed, smarty pants, higher rates of growth of aerobic bacteria. Don't skip the word aerobic. Let's think about that. Aerobic bacteria. So when you do aerobics, like in gym class, you're supposed to be breathing in a lot of oxygen. Aerobic means oxygen or oxygen needing or oxygen using. Anaerobic, the prefix an means without. So anaerobic bacteria prefer to be without, or they do best without oxygen. Aerobic bacteria need oxygen. Now, it's important that I point out here that oxygen is a byproduct or a waste product of photosynthesis. So if there's a lot of photosynthesis, the bacteria, the aerobic bacteria, they're going to be happy. Okay, let's keep looking. So it says he observed higher rates of growth of aerobic bacteria on algae grown in blue and red wavelengths of light. So let's take a look at this diagram. In the diagram, the dots or the little squiggles, those represent bacteria. And the higher number of bacteria we have, the more bacteria have been growing. Notice that there's a dip in how many bacteria are growing, and this is in the green part of our spectrum of the visible light. So we grow lots of bacteria here. So they're growing on algae. Algae is photosynthetic. So they're growing on top of the algae, and we get more of the bacteria when the algae is being grown in blue or purple light, and we get more of the bacteria when the algae is grown in red light, but we don't get very much of the bacteria when the algae is being grown in green light. And that's because the algae aren't photosynthesizing. They're reflecting that green light away. It's like he shined a green light bulb on the algae, and they're just sitting there going, no, can't do anything with that type of light. It just reflects away. And so then the bacteria don't grow because there's not any oxygen to feed them from photosynthesis. All right, hopefully that made sense, because you can almost guarantee AP is going to ask you a question about that. All right, so now we're going to talk about light entering the chloroplast, and I want to give you the big picture that the author of this particular presentation does, and it's very similar to what they did for the cell respiration presentation. So I want you to notice the orange box on the outside and then the blue box just on the inside. Those represent the two membranes of a chloroplast, the outer membrane and then the inner. The blue is the inner membrane. And then there's a green box. The green box represents one of those little green circles, the thylakoids. So it looks like a little coin, and you have a stack of them called the granum or grana. if you have several of them. So that's what that box will, so what's going on in that green box is happening in the thylakoid. What's happening in the blue box, that's the inside of the chloroplast, and the liquid is called the stroma, and you can see the word written down there. So that's the stroma. So I'm going to talk in this first set of notes just about what's going on within the thylakoid, or in the thylakoid membrane, actually. And then I'll pause and we'll do the next video and we'll talk about the Calvin cycle and finishing up photosynthesis in the second half. Okay, so remember that cell respiration was about breaking down glucose. So breaking it down, that makes it so cell respiration, breaking down, that's a catabolic process, and it is exergonic because it is releasing energy. Photosynthesis is exactly the opposite process. We're trying to collect energy and save it. And so that is an anabolic process we're putting together. And I want you to kind of think it through. We're taking, I shouldn't say we, plants, our benefactors, are taking our carbon dioxide out of the sky, and putting it together to make glucose molecules. So you would need 6 carbon dioxide molecules to make C6H12O6. So it's taking carbon dioxide, putting them together. That makes this an anabolic process, and it requires energy. Energy has to be put into the system in the form of light in this case, in order to do that. And so that's an endergonic process. Instead of giving off energy, it's taking in energy. The other thing that you need to be clear on is this whole idea of reduction and oxidation, because it come back and bite you in the tushy if you can't keep track of it. So oxidized things are giving away electrons. Reduced things are absorbing or taking on electrons. So in this equation here, the carbon dioxide is taking on electrons, and pretty much always when an electron gets taken on, so does a hydrogen. So that's where this hydrogen comes from. So it's bringing on hydrogens and electrons, and that makes it reduced. And oxidized means that it's giving up its electrons. So the water gets oxidized, and when it loses electrons, it usually loses its hydrogens also. So that's why it ends up becoming oxygen. So it goes from being water into being just plain oxygen. All right, so let's talk about when light enters, what exactly is going on. So I already talked about this part, I guess. I forgot this was here. Chloroplasts consist of a series of membranous discs arranged in stacks called grana. The grana are the inside of the inner membrane of the chloroplast. The stroma is the liquid. Oh, one part that I didn't mention that is important. If you have a photosynthetic prokaryote, so a bacteria that is able, or a protist that is able to do photosynthesis all by itself, then it does it on the folded membranes of the organism itself. So it might not have a chloroplast, it just does it on the inner membranes of the organism itself. All right. And then this is a nice diagram because it shows you the huge big picture of photosynthesis, sort of from beginning to end. So there's 2 main parts. There's the light reactions, which require light, and then don't ever call them the dark reactions. There's the Calvin cycle, and that part happens second. The light reactions occur in the membranes of those green thylakoid discs. Light is used to drive the production of ATP, so we just make a little ATP. I'm going to come back to that. And NADPH, the P for photosynthesis. Not really, it's for phosphate, but it might help you to remember photosynthesis, which is an electron shuttle. Now, let me just pause for a second and say, well, KJ, if we're making ATP and NADPH, why do we need to make glucose? If we've already made the energy, why are we making glucose? We're making glucose because ATP is a short-term energy molecule that is less stable. So that energy escapes easily. Glucose is more stable, so the plants are trying to give themselves energy for later, and so they're storing it in the form of glucose. Okay, and then NADPH is an electron shuttle, just like from cell respiration. We had FAD and we had NAD+. So this one is very similar. It's called NADP+, which is annoying. It's a similar electron shuttle, but not the same. I like to help students remember that, in that the P, the one that has the P in it, is the one that's in photosynthesis, which also starts with a P. All right, so the ATP gets made and the NADPH captures some electrons, and then those get brought over to what's called the Calvin cycle, named after the dude that discovered the Calvin cycle. And of course, he won an award. This occurs in the stroma. The Calvin cycle happens in the stroma. The ATP and NADPH are I expected it to say something different than it did. So they are produced in the light reactions, and they're used to make three. I'm trying to get this me out of the way. A 3-carbon sugar, which then gets put together to make the 6-carbon sugar of glucose or other carbohydrates. All right, moving on. So let's talk about when light enters into the thylakoid membrane, and that's what we're going to look at now. So here is an example of a chlorophyll molecule and what happens. So when a photon of light hits a chlorophyll molecule, and this is true for other atoms also, and molecules, but we're just talking about chlorophyll right now, an electron gets ejected. Remember our box chart, it gets ejected to a higher energy level. When it returns back to its neutral energy level, it releases a photon of light. So I think this is interesting. This is chlorophyll. It's a jar of or a beaker, a flask of chlorophyll that has had light hit it, so it's been lit up with photons. And now, as its electrons are going back down to their neutral state, it's releasing photons, interestingly, in the color that they absorbed. So the photons are red, and so they're releasing red photons. Could be blue also, could be a combination of the two. But noticeably, it's not green because it didn't absorb green. Okay, so it says when photons of light interact with chlorophyll, the electrons in the magnesium atom become excited. This happens with 1% of all sunlight that strikes the surface of the earth. Isolated chlorophyll will fluoresce when exposed to light as the excited electrons return back to their ground state. All right, so now we're going to take a look at the photosystems themselves. So this is sort of a generalized photosystem, and then we'll see the two put together. One thing I really like about your textbook is that they always use the same colors, and so the images go from one chapter to the next, and they're very understandable in that way. So take a look at the diagram. I want you to see the phospholipid bilayer. Do you see the lipids? Do you see the polar heads and the phospholipid tails? I didn't say that right at all. The lipid tails, oh my gosh. So that's the phospholipid bilayer that's part of these green little thylakoid membranes. Embedded in the membrane are these proteins, and your book always draws them purple. It always draws the proteins purple, so that's a way of you identifying them. Within these proteins are accessory pigments and different types of pigments. So they are represented by these green discs. So there's all kinds of pigments. Photons hit the different pigments, and the energy is transferred from one pigment to the next. As one electron gets excited, the next electron gets excited, the next electron gets excited, and energy is getting transferred from one to the next until it finally gets focused into what is called the reaction center complex. And in the reaction center complex is a special pair of chlorophyll A molecules. When their electrons get excited, they get ejected and actually lost. So they get ejected into what's called the primary electron acceptor. So usually electrons get raised up and then they fall back down, and they get raised up and they fall back down. In the case here, the magnesium atom that's in the chlorophyll ejects its electrons so far that they don't come back. Now that's a problem for magnesium because now it's missing electrons. So now we're going to have to replace those electrons with water. Water is going to come by and donate its electrons. That's going to cause water to fall apart, and we're going to be left with oxygen and some hydrogen ions. I'm jumping ahead, so let's take a look at what it says about photosystems. Photosystems are complexes of protein and pigment molecules that are embedded in the thylakoid membrane. So there's the thylakoid membrane, and there's the protein. They direct incoming photons to the reaction center. This lighter purple is the reaction center, where chlorophyll A molecules produce excited electrons, which are transferred to an electron transport chain. So they eject their electrons to this primary electron acceptor, and then it's going to go through an electron transport chain. We saw something similar in cell respiration. There are two types of photosystems. They are numbered awkwardly because they are numbered in the order in which they were discovered, which was backwards from the order in which they are actually used in photosynthesis. So photosystem 2 is what's activated first. And it has its central chlorophyll works best at a light wavelength of 680 nanometers. So it's called P680, and it's found at the beginning of the electron transport chain. Photosystem 1 is activated second. That's annoying. And its central chlorophylls work best at the wavelength of 700 nanometers, which is P700, and it's found at the end of the electron transport chain. That'll make more sense to you in just a minute. All right. Since chlorophyll, I'm back over on the reading here, since chlorophyll is not going to have electrons returned to it, new electrons are needed. I already told you where those new electrons are coming from. Water provides the replacement electrons. This is called photolysis. So photo refers to light, and then lysis, we're breaking something when we lyse it. So photolysis is happening to water and breaking the water up. So the water, H2 and 1O, donates its electrons, it loses its hydrogen ions, and then it's just a single O. Well, you can't have a single O. Oxygen is incredibly reactive. So it immediately finds another O, they covalently bond to each other, and you have an O2 molecule. So what that means, however, is that you have to have two water molecules to form the O2 molecule. Not a big deal, but so this creates 4 protons, 4 hydrogen ions, 1 molecule of oxygen gas, O2, for every 2 water molecules that are consumed. The oxygen gas is released as a waste product, becoming a major input for aerobic cell respiration. Good stuff. Okay, and now we have another chemiosmosis, and I like this diagram, and I don't know where to put my picture, but we have chemiosmosis again. If you remember that from cell respiration, that's where we had hydrogen ions that had a high concentration inside of the mitochondria, inside of the intermembrane space. So in between the two membranes, there were lots of hydrogens, low pH, acid, and they got pushed through ATP synthase, and that's what made ATP. Same identical thing is happening here for the same reason. We're trying to make some ATP, which is weird because ultimately we're not trying to make ATP like we were in cell respiration. Ultimately, we're trying to make sugar. It'll make sense in a minute. All right, so it says here, as the electrons move through the electron transport chain, they provide the energy for chemiosmosis or chemiosmosis. I always heard it chemi, and now recently I've heard chemi. Sorry. In a fashion almost identical to that in cell respiration. Protons are pumped by the electron transport proteins from the stroma into the thylakoid space. So look at this diagram. We'll look at it up close in just a second. So lots of hydrogens in there. The facilitated diffusion of protons, here that is, coming out. So it's high concentration to low concentration. No energy needed. In fact, just the opposite. This is diffusion. And it's allowing energy to actually be made because it's turning through this ATP synthase turbine that attaches a phosphate to ADP to make ATP. So through ATP synthase drives the synthesis of ATP. One notable difference, these are ugly words, ladies and gentlemen. In respiration, which is what we talked about last time, the energy comes from the oxidation of glucose, so it is called oxidative phosphorylation. So when we break down glucose and we donate all those electrons, we are oxidating it, and so it's called oxidative phosphorylation. Phosphorylation is when we're adding a phosphate to ADP. We are phosphorylating it. So that's called oxidative phosphorylation. However, this time we didn't have glucose. We're trying to make glucose. So this time when we are making ATP, the process is called photophosphorylation. That's because the energy source is from light, not from sugar. All right, so let's take a close up. This is the light reaction, the heart of the light reactions that you need to know. So here's Photosystem II. It's the first one, I know. And here's Photosystem I. So let's watch what happens. A photon of light hits the photosystem II, and it excites electrons in all these different pigments, and they pass the energy from one pigment to the next until it reaches the reaction center where the chlorophyll A's are, and the magnesium atom ejects its electrons, and the electrons are accepted as part of this electron transport chain. That means that they are missing electrons, so water has to donate its electrons. So we see the breakdown of water here and the formation of 2 hydrogen ions and a molecule of oxygen, O2. So what happens to those electrons? Those electrons, just like with cell respiration, they start at a high energy level, and very gradually, these are like the spark plugs in our gas tank. They're very gradually releasing their energy, and they're using that small amount of energy to do work. And in this case, the work is to pump hydrogen ions from outside to the inside of the thylakoid space. Remember from biochemistry that when you have lots of hydrogen ions, you have low pH. That makes it acidic on the inside. We'll come to that, back to that in just a moment. So now, once the electrons have hit their lowest energy level, let me say one thing and we'll come back to them. Again, a light photon hits the photosystem 1. The energy gets transferred from one pigment to the next until it gets to the central chlorophyll molecules. The magnesium atoms again completely eject their electrons, and now they're missing electrons. Well, instead of using water again, we just use the low energy electrons from the electron transport chain to replace their missing electrons. So the electrons are coming from water for photosystem 2, but they're coming from the electron transport chain for photosystem 1. So where do those ejected electrons go? They ultimately get taken by an enzyme that's called NADP plus reductase and given to NADP plus. So those electrons are added. Remember, hydrogen ions always come with because they're negative and positive and they're attracted to each other. So we end up with this electron shuttle called NADPH, and it's going to carry the electrons over to the Calvin cycle in the stroma. In the same breath, we have all these hydrogens that are collecting in the thylakoid space. They start diffusing through ATP synthase, and we start making a small amount of ATP. Now, remember, that's counterintuitive because you're thinking in photosynthesis, we make sugar, we don't make ATP. Well, we make a little bit of ATP, and then we turn it into glucose so that we can store that energy. ATP is short-lived. We don't want to be storing energy in ATP. All right, so we are getting near the end of this one. Just comparing the mitochondria and the chloroplast. Remember the endosymbiotic theory that they were both probably bacteria at one time, very similar in their structures. So look what's happening, and I've already made a lot of these comparisons. So the left side, imagine that this is the mitochondria. We had an electron transport chain, which was a whole bunch of proteins that were allowing the electrons to transfer, and they were pumping hydrogens into the intermembrane space between, remember the cristae, the folds of the mitochondria, the inner mitochondrial membrane. So it's in this dark gray area. That's where the hydrogen ions are collecting. Then they diffuse, those hydrogen ions diffuse back through and turn the turbines of ATP synthase to make ATP. And in cell respiration, we produce a ton of ATP. In the chloroplast, in these light reactions, we're doing something very, very similar. So imagine that this is the thylakoid membrane. We have an electron transport chain. As the photons hit, we're ejecting electrons. They're traveling through an electron transport chain. But this time, they're pumping the electrons into the space within the thylakoid, into the thylakoid space. And then the hydrogen ions are diffusing back through ATP synthase. They're turning the turbine, which allows the ADP and phosphate to join to make ATP. So we temporarily have some ATP. All right, and then this is another image, which I think is a stupendous image. It's really worth your time to look at, but it basically says what I've just said in the previous diagram. Here's the light. It's traveling through the different pigments. The electrons get ejected. Water breaks up. It decomposes and donates the electrons. The electrons travel through the electron transport chain, and some ATP gets made in the process. They again get ejected. Here's some more light coming in. So electrons again get ejected, and then they get captured by NADPH, and the NADPH carries them off. That's called non-cyclic electron flow, because we keep getting electrons from water and we keep sending them off somewhere else. We get electrons from water and we send send them off somewhere else. There is another thing, whoops, we're not going to look at that. There's another thing that's called cyclic electron flow, and this happens, it's super inefficient, and it happens if there's not enough water available for the plant. And it allows the plant to make ATP. Remember fermentation when we didn't have oxygen present? This is what happens in a plant if there's not enough water present. Doesn't allow it to store any of the energy. Very inefficient system, but it doesn't require any water. All right, and then I also think that this is a super cool diagram worth looking at. I think it's on page like 165 of your textbook, but it's a good way to kind of an analogy for students. So imagine that a photon of light, that's your energy, that's the energy that this construction worker hammers into the teeter-totter, and it ejects this ball, which is representing an electron. He's lost it. Now that electron is gone. It's been captured by somebody else. So that original construction worker no longer owns that electron. Water will have to replace his electron. So then she lets go of the electron downhill, and the electron is losing energy as it goes down, and it's being used to turn, actually in this analogy, it's being used to turn a turbine, which is cool because ATP synthase is also a type of turbine, and we're making a little bit of ATP. As the electron finally finishes off and is its lowest energy, another photon ejects it and causes the electron to be captured by somebody else, who then donates the electrons to NADPH, who then shuttles them over to the reaction, the Calvin cycle, which is taking place in the stroma. Fun fact, even if you are growing plants indoors, you are still using sunlight to do it, just a version that was stored in the chemical bonds of the fossil fuels that are being used to power your electric lights. Things that go into the light reactions and things that come out of the light reactions. Obviously, light goes into the light reactions. ADP, an inorganic phosphate, NADP+, and water go in. What comes out? You have ATP, NADPH, and some oxygen. This is where the oxygen is escaping out of the chloroplast. It's being produced as a waste product, but it's going to be super important for the mitochondria. Now I'm going to go ahead and pause. We're going to talk about the Calvin cycle for the next set of notes.